Hello guys, welcome back to our channel. So this is the part 2 of Cell the Unit of Life chapter, and so let's start reading. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasm and PPLO means pleuropneumonia-like organisms. They are generally smaller and multiple more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and psyllium, spiral. The organization of the prokaryotic cell is fundamentally similar even though prokaryotes exist. All prokaryotes have a cell wall surrounding the cell membrane except the mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition, genomic DNA, the single chromosome circular DNA. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmids DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotic. In higher classes, you will learn that this plasmid's DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. The outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the difference in the cell envelope and the manner in which they respond to the staining procedure developed by Gram, which those that take up the Gram stain are Gram positive and the others that do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheet called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shapes of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. Mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamina. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides, flagella, pili, and fimbriae are also surface structure of the bacteria but do not play a role in motility. The pili are elongated tubular structure made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissue. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together from 70S. Prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophycine granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and are large central vacuoles which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plants. So let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enable the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensured that the nonpolar tail of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipids, membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. Depending on the ease of extraction, membrane protein can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model. According to this, the chaotic fluid nature of lipid enable lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junction, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on their side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion or along the concentration gradient from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis, as the polar molecules cannot pass through the nonpolar lipid bilayer. They require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. Example, sodium-potassium pump. Cell wall, as you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose, gelatin, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth which gradually diminish as the cell matures, and the secondary. We will continue with part 3. Thank you for watching.